Hi, I'm Erika Dudkin from the University of Bonn, and I will present to you the distribution-free differential expression analysis for single-cell RNA sequencing data across patient groups. So first of all, what is differential expression analysis? This deals with finding the genes which are significantly differentially expressed between two samples or two groups. And for the standard setting where we are comparing one sample versus another sample and want to find the differentially expressed genes, already many methods exist also for the single-cell RNA sequencing setting. But what if we want to compare multiple patients from one group versus multiple patients from another group and want to find there the genes that are significantly differentially expressed between the two groups, and we want to also preserve the patient intervariability within one group. So this project started with our collaboration partners, Kevin Basler and Joachim Schulze, where we wanted to investigate the chronic obstructive pulmonary disease. And we wanted to find the gene level differences between a healthy patient cohort and a COPD patient cohort with single-cell RNA sequencing data. So how can we find now the differentially expressed genes in this multi-patient setting? One easy or standard method we could do is just calculate the mean for each patient for each gene within the group and then compare the means with RNA sequencing differential expression analysis methods. But then we would lose all the single-cell precision, single-cell resolution precision. That we didn't want to do, so we came up with our own method. And how does it work? So first of all, we are performing a pairwise Wilcoxon rank sum test between each patient from one group versus each patient from the other group. We are collecting all the Wilcoxon scores and calculating then the median out of all Wilcoxon scores, which is then our test statistic. How are we then evaluating how significant this test statistic is? There we are performing a permutation test, so we are shuffling one patient from one group to the other group, and this we are doing with all possible permutations and combinations. And for each permutation, we are calculating again the pairwise Wilcoxon rank sum test, calculating the median out of all Wilcoxon scores, and then with all different permutations, so we get our null distribution and then can calculate our p-value. So with that, we get all the genes that are significantly differentially expressed or not. So we get a list with all the genes, and that is then our result. But how does this newly proposed method perform? So we also evaluated the performance of the differential expression analysis method while simulating different single-cell RNA sequencing data sets. We were looking at two different cases for now. Once we simulated the different distributions between two groups, which has similar mean, which is shown here in the black line. So the mean between the two groups and the patients is similar, but the distributions between the two groups is different. And we wanted to show that our method is able to find these differences in the distributions, although the mean is similar, which would be not possible with methods which take just the mean of the single-cell RNA sequencing data. And on the other hand, we simulated cases with same distributions between the two groups in order to show that our method can also control the false positive rates. Just shortly to the results, we compared it to HR, pretty well-known differential expression analysis method, which considers the mean of the single-cell RNA sequencing data. And here we can see the false negative rates. So our method, which is the blue one, performs for most cases pretty good for the false negative rates. So the different distributions were identified between the two groups, whereas HR could not find the differences here shown with high false negative rates. And on the other hand, with the same distributions, both methods perform equally well with low false positive rates. So thank you for your attention, and if you have any questions, I'm happy to answer them. Thanks.